Usually when we talk about our immune system detecting a threat, there is this expectation of ruthless mechanical precision. A pathogen enters, your immune cells' surface receptors act like, a barcode scanner. They scan the threat and the alarm goes off. It feels clean.
Right. It feels absolute. It's comforting to think of it that way because the classic textbook model of immunity is very binary. Threat detected, response initiated.
But then you step into the world of intracellular parasites, specifically how human cells detect Toxoplasma gondii, and suddenly that barcode scanner is, well, it's completely useless. We're looking at an immunological landscape that is murky and just incredibly complex.
Extremely complex, yeah.
And that brings us to the mission for today's deep dive. Welcome to everyone listening. Today, you and I are looking at a brand new 2026 paper from Cell Death and Disease that basically cracks this mystery wide open. It outlines in staggering detail exactly how Toxoplasma hacks our macrophages, our immune first responders, to trigger severe inflammation.
It really is a masterpiece of molecular detective work because for a long time, the central question has been about our biological radar. So mice, for example, have these receptors called TLR11 and TLR12.
Okay.
And they serve as this built-in, highly sensitive Toxoplasma alarm system. But humans, we don't have functional versions of those receptors. We lack the early warning radar entirely. So how on earth do human cells know this widespread parasite is even there?
Which implies one of two things, right? Either the parasite is operating in total stealth mode, or our cells are using a completely different tripwire.
Exactly.
And what this paper reveals is that it's definitely not stealth. The parasite actively breaks into our macrophages, rewires their cellular brains, completely alters their diet, and forces the host cell to build it a safe house. And then as a final act of sabotage, it blows the house up to spread.
It's a hostile takeover at the most microscopic level. The parasite isn't just hiding, it is micromanaging the host's own survival mechanisms to ensure its own replication.
Okay, let's unpack this. We need to understand exactly how a microscopic parasite gets its hands on a macrophage's steering wheel right after it enters. Because this isn't just, a pathogen floating in the bloodstream. It has to breach the cell membrane and take control of the internal machinery.
Right. And the initial invasion relies on what I'd call a masterclass in biochemical engineering. So the parasite utilizes a specific protein called ASP5. This is a Golgi resident aspartyl protease.
Okay, ASP5.
Yeah, and if we step away from the jargon for a second, think of ASP5 as a highly specialized molecular crowbar. It's an enzyme the parasite uses to pry open a communication channel and secrete a package of factors known as TGESPs.
And that stands for T-gondii excretory and secretory proteins, right?
Exactly, TGESP. So it drops these secretory proteins into the host cell via exosomes. And once those parasite proteins are floating around inside our macrophage, they don't just randomly cause damage.
They have a target.
Right. They specifically target a host signaling cascade. The paper focuses heavily on the PI3K and AKT pathway, which eventually leads to a protein called vt-catenin.
Okay, so for everyone listening, a good way to remember this is to think of the game Pac-Man. Pac. So P for PI3K, A for AKT, and C for catenin.
I love that. Pac, yes. That cascade is central to the host cell's communication network. What the parasite's secreted factors do is trigger the activation of PI3K. And in the molecular world, activation usually means phosphorylation.
You're adding a phosphate group.
Exactly. You add a phosphate group to a protein, which physically changes its three-dimensional shape and turns it on. The parasite triggers the phosphorylation of PI3K, specifically at a spot called tyrosine 4-phosphate.
Tyrosine 4-phosphate, got it.
And that activated PI3K then acts as a kinase itself, moving down the chain to phosphorylate AKT at serine 473.
So P activates A, and AKT acts as the final messenger that phosphorylates the C, catenin.
Yes, exactly.
But I have to stop and look at the audacity of this. Wait, why would an invading parasite want to turn on the host's own cellular pathways? If you break into a highly secure facility, the last thing you want to do is, start flipping the internal breakers and turning on the smart home systems.
Well, unless you know exactly how the smart home is wired, right? You want to
Yep.
lock the doors and disable the internal cameras. The parasite isn't triggering a random pathway. It is incredibly surgical. When AKT phosphorylates vt-catenin, it does so at one very specific amino acid, serine 552.
For 552.
Right. That single molecular modification acts as a VIP pass.
A VIP pass to the nucleus. Because normally vt-catenin is structural, or it's just, hanging out in the main body of the cell, the cytoplasm.
Yeah, it's just waiting around.
But with that specific phosphate group attached to serine 552, the cell's internal security recognizes it as highly important and shuttles it straight into the nucleus, which is the control room where all our DNA is stored.
Precisely. You see, the parasite doesn't have the tools to rewrite the host's genetic code from the outside. It needs a Trojan horse. By modifying the host's own β-catenin, it sends a trusted messenger into the control room to start pulling levers on its behalf.
So the burglar uses the homeowner's own fingerprint to unlock the safe. That is wild.
It really is.
Okay, let's pause for a quick recap of the first 5 minutes for you listening. T. gondii enters the macrophage. It uses the ASP5 crowbar to drop secretory proteins, and those proteins activate the PK pathway, PI3K, to AKT to β-catenin, which sends β-catenin into the nucleus. So I want you to think about this. If you were designing a drug right now, where would you cut this wire? At P, at A, or at C? Keep that in the back of your mind.
That's a great question to be thinking about as we move into the next phase.
Right, because that brings us to what happens once β-catenin is actually inside the nucleus. It's in the control room, surrounded by our DNA. Now, the paper details this massive cascade of transcription factors, proteins that turn genes on and off.
Right, the alphabet soup.
Yeah, exactly. The alphabet soup of proteins like TCF4 and IRF4. But instead of getting lost in all those letters, what we're really looking at is the parasite using β-catenin to turn on an inflammatory factory, right?
That's the perfect way to look at it. So phosphorylated β-catenin binds with a transcription factor called TCF4. Together they drive the transcription of a gene called IRF4.
But IRF4 needs its own activation, doesn't it?
It does. A kinase called ROCK2 has to come in and phosphorylate IRF4. And once that happens, activated IRF4 acts as a transcription factor for another gene, which is the ultimate target here, a gene called CYBB, which immunologists also know as NOx2.
CYBB.
Right.
And CYBB is the blueprint for an enzyme complex whose primary job is to generate reactive oxygen species, or ROS.
Exactly, ROS.
And we hear a lot about ROS and oxidative stress in pop science, usually in the context of why we should eat blueberries and antioxidants. It's essentially molecular smoke. It damages proteins, it damages lipids, it's chaotic.
Yeah.
So if I'm tracking the parasite's logic here, it hacks the nucleus specifically to force the cell to churn out massive amounts of toxic molecular smoke.
That's what happens, yes.
But why? Why fill your brand new safe house with toxic smoke?
It seems totally counterintuitive until you look at the cell's internal layout. Early in the infection, we're talking maybe 6 hours in this burst of ROS, specifically stresses the host cell's mitochondria.
Power plants of the cell.
Right. And they are very sensitive to oxidative stress. When they get damaged by this smoke, the cell's automated maintenance systems kick in.
The cleanup crew.
Exactly. A process called mitophagy. The cell uses a sensor system proteins called pulkk1 and parkin to detect the damaged mitochondria and tag them for destruction and recycling.
Wait, so the parasite starts a localized fire specifically to force the host's internal cleanup crew to demolish the cellular power plants.
It is quite literally bulldozing the furniture. By intentionally triggering this early mitophagy, the parasite forces the host cell to clear out vast amounts of physical space in the cytoplasm. It creates this permissive, empty environment. And in that newly cleared space, the parasite builds its parasitophorous vacuole, the PV.
The nursery. That is just sinister.
Yeah.
It uses our own garbage disposal systems to clear a massive plot of land so it can build a protective bubble where it can replicate safely, shielded from the macrophage's normal weapons.
It's a masterclass in exploding host infrastructure. The evolutionary arms race here is staggering. The macrophage exists to consume and destroy pathogens, but the pathogen has evolved to get consumed, hijack the brain, trigger a controlled burn, and use the fire department to build a luxury suite.
Here's where it gets really interesting. Let's do our second recap. So nuclear catenin pairs with TCF4, which turns on IRF4, activated by ROCK2, which finally turns on CYBB. And CYBB makes massive amounts of ROS smoke. The ROS triggers mitophagy via PINK1 and parkin to clear out damaged mitochondria so the parasite can build its nursery.
That's the sequence.
So for everyone listening, can you see how the parasite is using our own cellular cleanup crew to build its nursery? It's incredible. But a controlled burn only works if you can keep it controlled.
Right, which is the catch.
And according to the paper, as we move later into the infection timeline, around 12 to 30 hours, that dynamic completely shifts.
Right.
The parasite has replicated, the nursery gets crowded, and eventually it has to get out. It has to flip the switch from this cozy safe house to a controlled demolition.
And the brilliant part is it uses the exact same mechanism to do both. That molecular smoke machine, CYBB, The parasite never turned it off. It just keeps churning out ROS.
Oh, wow.
Yeah. So eventually, the oxidative stress becomes too overwhelming for the mitophagy cleanup crew to handle. The remaining mitochondria don't just get recycled safely. They begin to physically fragment and disintegrate.
And when a cellular power plant disintegrates, it spills its contents everywhere. Which brings us to a really fascinating biological tripwire. When the mitochondria burst, they release their own DNA, mitochondrial DNA, or mtDNA. But because of all that ROS smoke the parasite generated for hours, this isn't normal DNA anymore. It's highly oxidized, and it's suddenly floating around in the main body of the cell where it absolutely does not belong.
This is where the host's innate immune system finally wakes up. You have to remember the evolutionary origin of mitochondria. Billions of years ago, they were likely free-living, ancient bacteria that were engulfed by larger cells.
Right, the endosymbiotic there.
Exactly. Because of that, mitochondrial DNA is circular. It lacks certain protective proteins that our nuclear DNA has. To the cell's sensors, it looks bacterial.
So when oxidized, bacterial-looking DNA suddenly floods the cytoplasm, the macrophages' internal sensors don't just see cellular damage, they see a massive catastrophic infection. It's a host-associated molecular pattern, AMP, the ultimate alarm bell.
Yes. The cell has a specialized receptor called AIM2 that is designed specifically to detect this misplaced oxidized DNA. The moment AIM2 binds to it, it initiates the assembly of a massive multi-protein complex called an inflammasome.
The AMM2 and NLRP3 inflammasome. It teams up with a protein called ASC, right?
Correct. ASC acts as the glue.
And an inflammasome is essentially a biological weapon. It recruits an enzyme called caspase-1. Now, textbooks often describe caspase-1 as, molecular scissors that just float around cutting things. But in this context, that feels way too gentle. When this inflammasome forms, caspase-1 is more like pulling the pin on a grenade.
That is a much more accurate visualization of the stakes. Caspase-1 activates a highly potent inflammatory molecule called IL-1, which gets secreted to basically scream for backup from the rest of the immune system. But its most devastating action is acting on a protein called gasdermin D.
The shrapnel.
Yes. When caspase-1 cleaves gasdermin D, the protein uncoils and physically embeds itself into the outer membrane of the macrophage. Dozens, hundreds of these proteins group together and punch giant, gaping holes through the cell's outer wall.
Which completely destroys the cell's ability to regulate its internal pressure. Water rushes in and the macrophage literally bursts. It's a highly inflammatory, fiery death called pyroptosis. What's wild to me is that in all this destruction, the nucleus usually stays intact, but the outer membrane is completely shredded. And where are the newly replicated parasites during this explosion?
Sitting perfectly unharmed.
Right. They're in their protective bubble, just waiting for the walls to come down. Once the cell bursts, they just flood out through the holes into the surrounding tissue to infect fresh, healthy cells.
Wait, so the parasite intentionally triggers a massive inflammatory explosion just to hitch a ride out of the cell?
It does. The parasite perversely uses the host cell's ultimate kamikaze defense mechanism as a getaway car. It intentionally pushes the cell toward an inflammatory explosion because that explosion is the fastest, most efficient way to disperse its progeny.
Okay, time for recap number 3. Excess ROS causes oxidized mtDNA to leak out. The AIM2 and NLRP3 inflammasome detects it, activates caspase-1, which cuts gasdermin D, leading to pyroptosis and the cell bursting. So think about this for a second, how oxidized mitochondrial DNA acts as the ultimate biological tripwire. What other diseases might involve this same tripwire? It's a fascinating thought. It really challenges our assumptions about pathology. Are the symptoms of an infection the result of our immune system fighting back, or are they the result of the pathogen pulling our strings to facilitate its life cycle? In the case of Toxoplasma, it's definitely the latter.
And as if manipulating the cell's death wasn't enough, this paper dives into a parallel manipulation happening at the exact same time. While the parasite is building inflammasomes and loading up the gasdermin D shrapnel, it's also fundamentally changing the macrophage's diet.
Right, the metabolic hijack.
Yeah, it alters the cellular metabolism to fund this entire bomb-making effort.
This is perhaps the most insidious part of the takeover. It all traces back to that early ROS smoke and our hijacked VIP messenger β-catenin. The oxidative stress generated by the parasite does something else in the background. It stabilizes a host protein called HIF-1A.
And HIF-1 is a master metabolic switch. Normally, if a cell has plenty of oxygen, this protein is rapidly sent to the cellular trash can. It's only supposed to be active when oxygen is incredibly low, like if you're exercising heavily and your muscles need anaerobic energy. But the parasite's smokescreen artificially preserves it.
Exactly. By preventing its destruction, the parasite forces HIF-1 to remain active. And this protein marches into the nucleus and completely rewrites how the macrophage processes energy. It turns on enzymes like HKAI and LDHA.
And what does that do to the cell? Because a normal resting macrophage uses a highly efficient process called oxidative phosphorylation, the TCA cycle, to make energy. It's clean, it's sustainable.
But HIF-1 throws all that out the window. It forces the cell to start rapidly consuming glucose and churning out lactate, even when there's plenty of oxygen available. It forces the cell into aerobic glycolysis.
Which biologists immediately recognize as the Warburg effect. We see this all the time in cancer biology. Tumors shift to this exact same inefficient, sugar-hungry metabolism because it allows them to rapidly generate the building blocks needed for fast replication. And here, the parasite forces the immune cell into the Warburg effect.
It is force-feeding the cell sugar to push it into what immunologists call the M1 state, which is an angry, hyperinflammatory state. The macrophage starts pumping out inflammatory cytokines like IL-12, IL-6, and TNF-A.
And the paper notes something fascinating about that. This localized metabolic shift actually bleeds out into the surrounding environment and alters the behavior of other immune cells, right?
Yes, it's a ripple effect. The researchers found that T cells in this environment developed a broken TCA cycle, specifically CD4 plus T cells. They were forced to rely on glycolysis too, which fueled a massive damaging inflammatory response across the board.
So the parasite doesn't just hijack the host cell, it basically terraforms the local tissue environment. It ensures that the metabolic conditions are perfectly tuned to support the massive inflammatory explosion it needs to escape.
It is a dark, brilliant piece of biology.
But this deep dive isn't just a horror story about a microscopic hacker, because the researchers mapped every single step of this heist, from the ASP5 crowbar to the catenin VIP pass to the ROS smokescreen to the Warburg effect. They figured out exactly how to stop it. And this brings us to the rescue plan.
Yes, the therapeutic implications of this paper are just massive. Because they mapped the host pathways the parasite relies on, they realized they didn't need to attack the parasite directly. They could just shut off the compromised pathways.
Right. We can use drugs. They tested several inhibitors that target the PASI cascade, the pi3-catocatenin cascade. So they looked at copanlisib, which is a PI3K inhibitor, and capivasertib, which is an AKT inhibitor.
Both very effective.
But the undisputed star of the show was a compound called XAV939. This is an inhibitor that specifically targets enzymes called tankurases, specifically tankurase 1 and 2. How does hitting tankurase stop this entire cascade?
Well, tankurases normally protect pignin from being destroyed. By inhibiting them with XAV939, you essentially flag all that hijacked catenin for the cellular trash can.
Oh.
It undergoes ubiquitin-mediated degradation.
The cell shreds it.
The cell shreds it. And without AKT acting as the VIP messenger in the nucleus, the entire house of cards collapses.
It's beautiful logic. No catenin means the inflammatory factories are never turned on. CYBB never gets activated. There's no massive ROS smokescreen. Without the toxic smoke, the mitochondria stay healthy. The mitophagy cleanup crew never clears space for the parasite's nursery.
Exactly.
The oxidized DNA never leaks out to trip the AAM2 inflammasome. Gastromin D never turns into shrapnel. The entire explosion is canceled.
And metabolically, the cell naturally shifts away from the Warburg effect. It goes back to its efficient resting state. It enters an M2 healing phase rather than that angry M1 state.
And the parasite.
Because it can't force the cell to build its nursery or provide the energy for its escape, it is trapped. It cannot replicate, and it simply dies.
And the researchers proved this in vivo, right? They gave mice lethal doses of T. gondii, treated them with this XAV939 inhibitor, and the mice actually survived.
They did. It's a huge breakthrough.
But the detail that really highlights why this approach is the future of medicine is the molecular docking study. They use 3D computer simulations to see exactly how these drugs physically interact with the proteins.
This is where you see the sheer elegance of the strategy. They mapped the physical structure of the mammalian proteins, our PI3K, our tankurases, and compared them to the parasite's evolutionary versions of those same proteins.
Because parasites have their own internal signaling networks.
Right, exactly. When they model a drug like copanlisib, which targets PI3K, they saw that the drug fit perfectly into a deep, well-defined pocket in the mammalian version of the protein. It anchored itself using a dense network of hydrogen bonds with very specific amino acids. It was a perfect, rigid, lock-and-key fit.
But when they ran the same simulation on the parasite's version of PI3K, what happened?
The pocket was entirely different. It was wide open, shallow, and lacked the key acidic residues needed for those hydrogen bonds. The drug basically floated off of it. It barely bound at all. And the exact same dynamic was true for XAV939 and the parasite's version of tankerase.
So what does this all mean? The drugs bind tightly to the host machinery, but they completely ignore the parasite's machinery.
Right, which means this is a perfect example of host-directed therapy. We spend decades trying to invent new antibiotics and antiparasitics that poison the bugs directly. But pathogens replicate so fast, they mutate, and they eventually evolve resistance. They figure out how to pick the new locks we put on our drugs.
That is the crisis of antimicrobial resistance in a nutshell. We put immense selective pressure on the pathogen to evolve.
But host-directed therapy completely flips the paradigm. We aren't trying to poison the burglar, we're just temporarily unplugging our own smart home system.
I love that. The parasite breaks in, expects the lights to turn on and the alarms to disable, and instead the power is just out. It can't use our machinery against us, and it dies not from a poison, but from a total lack of leverage.
And because the drug targets our stable, slowly evolving human proteins, the parasite has practically no way to mutate around it.
It is a phenomenal synthesis of immunology, metabolism, and pharmacology. It proves that sometimes the best way to win a fight is to just stop giving your opponent the weapons.
I want to thank everyone listening for taking this incredibly dense microscopic journey with us. We tracked a parasite as it hacks the PAPE pathway, creates ROS smoke screens, blows up the cell with Gasdermin D, and changes the cell's diet through the Warburg effect. And the cure isn't a bigger gun, it's just changing the locks on our own doors.
The elegance of that solution is deeply satisfying. But I want to leave you with one final thought to mull over. We spent this time looking at how Toxoplasma uses this β-catenin pathway and the Warburg effect to survive and conquer. But these are fundamental biological levers. It raises a massive, slightly chilling question, which is how many others are doing this?
Exactly. If this pathway is so critical for a parasite to survive, how many other intracellular pathogens, bacteria, viruses, or even aggressive cancers are using this exact same playbook? They are turning our cellular factories against us, hiding in plain sight by using our own metabolic pathways as a shield. The next time you read about a localized, stubborn infection or a complex tumor microenvironment, ask yourself, is the disease actually overpowering the host, or has it just brilliantly tricked the host into defeating itself?
That completely changes how you look at the progression of any disease. We started this deep dive thinking our immune radar was just broken, faulty barcode scanner unable to see the threat. But it turns out the alarm system isn't broken at all. The intruder is just a master electrician using our own wiring to rig the explosives. Keep questioning the world around you, keep looking for the hidden wiring behind the headlines, and thank you for joining us on this deep dive.